So welcome to everyone who's joining this latest DRIIMB webinar. We're about ready to start, but we'll just give people a moment to settle in. Oh, I see our speaker has at least one fan in the audience.
Hi, Gary.
Has everyone come in immediately, or are they joining? Because we look to be fairly full, but are they still connecting? I can't tell.
Yeah, remember, yeah, it's raising, but very slightly. So I think we can start.
Well, I mean, I'll just give a general introduction while people get comfy and settled in. So welcome to this. This is the latest in a series of DRIIMB webinars. DRIIMB, as I say every time, is a consortium of different structural biology CCPs, collaborative computational projects. And DRIIMB is aiming to build capacity in integrative structural biology. So we're interested in building bridges between the different techniques and looking at making it easier to apply multiple techniques to particular biological systems. So as part of that, we have this webinar series exploring different approaches to structural biology. So this time I am very happy to welcome Steve Muench from Leeds, who's going to talk to us about time-resolved CryoEM this time. So Steve, I believe, was originally a crystallographer until 2005, when he saw the light and moved to Leeds and got into CryoEM. He's since then built up his own group looking at membrane proteins and drug design, sample preparation, and what he's going to talk about today on time-resolved CryoEM. We know him very well in CCP-EM. He's contributed to many events, and he was co-organizer of the 2023 Spring Symposium. At Leeds, he's involved in lots of stuff. I should particularly highlight that he's Deputy Director of the Asbury Centre there in Leeds. So in a moment, I'll pass over to Steve to give his talk. Before that, I just want to highlight that the next DRIIMB webinar is going to be by Dr. Shruti Viswanath on recent developments in integrative structural modelling of macromolecular assemblies, and that's going to be towards the end of August. So I guess we don't have an exact date yet, do we? But that will be announced soon.
Yeah, it's arranged like 27th of August, but I'll confirm the time soon.
Okay, 27th of August. Okay, there are details in the chat. You can find them there. If during the talk you want to ask any questions, please could you put them into the Q&A, and we'll cover them at the end of the talk. I think that's all I need to cover. So with no more waffling, please can I pass over to Steve to give us his talk. Thanks.
Great. Thank you, Matt, and thank you for the invitation as well. Let me just battle with the technology. Hopefully, there we go. Can everyone see that now? Is that okay?
Yep, that's great.
Right, now just a laser pointer. Brilliant. Okay, well, thank you. So, afternoon, everyone. So, as Martin's already mentioned, what I wanted to talk about today was some of the work that we've been doing over in Leeds around time-resolved electron microscopy, but also broaden it out towards the end about the role of some of the other sort of techniques as well that can really come into play around sort of single particle EM. And I'm going to talk primarily about my work, but I thought it was important as well to highlight the work of a few other people as well, because there's some really interesting developments in this field at the moment that hopefully might sort of appeal to a few people in terms of their structural biology and what they're trying to do at the moment. I think I was a crystallographer. I still dabble a little bit in crystallography, and I've dabbled in mass spectrometry and a number of different structural biology techniques. I think it's really important that to answer the big questions, you've kind of got to use more than one technique these days. I feel it feels like you've got to have this collaborative approach. So with that out of the way, I will start with time-resolved EM, and I'm contractually obliged to show the Muench horse when you talk about time-resolved methodologies, because it's sort of the classic example, really, of why we do these sorts of experiments. So if we want to understand the mechanism of an object, for example, a horse, we can get some insights from a static snapshot. So here we can see, for example, the number of legs, etc. But to really understand the mechanistic detail, what it's good to be able to do is take a series of snapshots of your molecule in motion. This then gives you an order of events and, importantly, a timestamp between those events as well. So when you put these things together, you can get this more information-rich sort of Mackle movie, which allows you to understand more of your favorite protein/complex than you can from the static view. And that's sort of traditionally how we describe these sorts of time-resolved experiments. So it's trying to capture these non-equilibrium states, which often happen in a very short time scale after the addition of ATP, for example. There's another quick example I was going to sort of lean into, which we don't use so often, and that's actually some of the things that we've been doing in the EM as well, whereas instead of just understanding an individual protein, for example, we can start to look at larger complex formation as well. For example, the formation of large protein complexes, or we've been working with materials as well and trying to understand sort of the formation of these larger macromolecular structures, which can be multi-component parts. So you can have a selection of Lego bricks, for example, and then it's trying to work out how they then come together to form these different larger complexes, and then trying to capture those intermediary states allows us to understand sort of the instructions, if you like, of how these things come and form together, and how when you go off pathway you might start to make something which is maybe not what you tried to do in the first instance. So it's about understanding dynamics, it's just understanding how proteins function, but also we're looking at how things come together as well in these larger scale sort of ways. In terms of the scales, primarily for time-resolved electron microscopy, we're looking at large domain motions. We're typically in the millisecond time scale. I'll show you some examples where we can go to microseconds as well, but that's really where the technology ends. But that's why it's so complementary to, for example, time-resolved crystallography, because of course time-resolved crystallography is an incredibly powerful technique for looking at sort of femtosecond, picosecond, microsecond changes in a protein in that sort of crystal environment. We can typically go, from maybe 50, 60 kilodaltons upwards, all the way into sort of mitochondria, looking at organelles, looking at bacteria as well. So I won't talk so much about the in situ, but we can use EM for sort of in situ time-resolved studies as well, typically on the longer length and time scale, but still important biological questions to be answered there. And many other techniques can really start to fill the hole for these much faster transitions. And of course, a lot of structural biology happens below 50 kilodaltons, which is where things like sort of X-ray crystallography, mass spectrometry, NMR, etc., can really come into play to look at dynamics in these systems. So in order to understand sort of how we do the time-resolved EM, I'm going to take a step back, and apologies for those people that do EM at the moment, but I'm going to very quickly describe how we go about making an EM grid and processing the data, because it's important to understand that, to understand how some of these new systems that we've developed and others work. So huge thanks to Oksana and EM Facility for lending me these next few slides. So we have protein in solution floating around. We then put this protein on a specialized EM grid, typically around 6 millimeters in diameter, and then we blot away the vast majority of this liquid to leave a very thin film, which we then plunge in liquid ethane to generate vitreous ice, which we can then image. And the grids themselves can be made up of sort of regular arrays or irregular arrays of holes as well. We typically use the regular array of holes, but I won't bore you with the details of that. It's not relevant for this talk. I will just quickly play this video. So this is a speeded up video of how we typically make grids on the VitraBot system. So the grid is at the end of the tweezers. We then add approximately 3 microliters onto the grid. as I mentioned, we will then go up and blot it with a blotting paper. This then removes sort of 99.9%. So we're left with a very, very thin film, few sort of 100 nanometers thick, which we then plunge into the liquid ethane. And this then produces this thin vitreous ice into which our protein resides, hopefully in various different orientations, so we get around the orientation problem. We then apply the electrons. This then gives us these electron micrographs, which are incredibly noisy. Even with the really good cameras that we've got now, we still don't have the averaging power that you have in, for example, X-ray crystallography. But one advantage we do have is that we can pick out the individual protein molecules. And now the image processing techniques have got so good that what we can start to do is to align particles that belong to the same 2D view. And then we can sort of get a big range of these different two-dimensional views. And at this point, we can often start to pull out different conformational states. And I'll sort of take a break at this point to say that one of the things that we often do in time-resolved EMs, it's not about capturing one particular state at one particular time point. What we often see is that over a series of time points, you'll see a particular state start to increase in population, another one will start to decrease in population. You might get a certain state that only exists in a certain time frame within the experiment. So it's often about looking at how populations increase and decrease over time to give you this idea of how the different timestamps come together. But computationally now, we're getting really good at pulling out these different conformational states, even in a bit of an ensemble population. Then we can put all these different two-dimensional views together, and then we can generate these beautiful three-dimensional maps, and then we can hopefully try to see, I was going to say atomic, but that's quite a powerful word, see side chains. Sometimes we can get to atomic resolution, but typically we get to high resolution where we can start to see side chains and waters and things like that, which are obviously important. So with that said, now we know how we grid, how does that relate to how we do time-resolved electron microscopy? So what I've got here are three of the traditional ways that we would go about doing this. So in A is the light triggered reaction approach. So here you would blot the grid as you would do on the VitraBot. The grid would then be in the protein will be in the thin film, and we can then expose the protein to a particular light source. And this would then allow you to decage a compound or directly trigger a light triggered reaction, for example. And then the reaction stops once you hit the liquid ethane. So this can work incredibly well, but of course it's dependent on having something that reacts to light / something that you can decage in an appropriate timescale. So another way in which we can do this is we can take our specimen grid, we blot it to get the thin film again, and then we spray something on the thin film. So for example, we might spray ATP onto the protein. As soon as the ATP hits the protein, we start to trigger the reaction, and then we can quench the reaction, stop the reaction once we've frozen the protein in the liquid ethane. So again, we can have time, which is dependent about how quickly we can spray on the grid and plunge it in the liquid ethane. And this can work incredibly well. The difficulties can be in the mixing, knowing exactly where you sprayed on the grid. You don't always get uniform spraying. So there are some limitations to this approach. So an alternative approach is to mix. So you could mix your protein with ATP, for example, in a microfluidic mixer, and then you directly spray this on the grid. You get excellent mixing, but the challenge here is that you have to have the droplets in such a way that when they hit the grid, they land and they spread out to form thin enough films that when you freeze them in liquid ethane, you can then image them in the microscope. And it's generating these droplets and these films, which is the real challenge here, although we get over the poor mixing with these approaches. And of course, you know, there's a history of this within the EM community, and one of the classic experiments is the Behrman and Ullmann experiment from 1994. So here you have some tweezers, a grid at the end. Here what they were doing was spraying reactant on the grid, acetylcholine esterase receptor, and then the grid was being plunged in the liquid ethane, which is just here. And I believe that this equipment has actually been dusted down in the last few years and used again in the LMB because, you know, fundamentally it works well. You spray reactant on the grid, a reaction starts, you end the reaction in the ethane. You don't need complex setups necessarily. When I started in Leeds, this is actually the equipment I trained on to make EM grids before we bought a VitraBot, and it was quite an interesting device. It was homemade, as you can tell. It had a gas cylinder, so it was gas powered. And what you would do is blot the grids at the top here. You would then spray a reactant on here if you wanted to, and then you plunge it in the liquid ethane, which you can see here. And so again, it's this spray approach. And this was done in collaboration really through Howard White and John Trinick. They'd really led the work on this. And I joined in around 2005 and started to work with Howard White. And Howard White was also working with Joachim Frank over in the States. And what Joachim was doing was, rather than directly spraying on the grid, he was doing this mixing approach. So mixing in microfluidics, then directly spraying on the grid. And he was doing that with Howard White as well. And we sort of incorporated this mixing approach into our design, which we sort of published in 2018. This is several iterations on from where we started. But there's a number of groups doing this. I don't have time to talk about them all, but there's a really nice setup over in the CRIC as well with the NCHEV group. They did a beautiful publication not long ago, and they're doing some really interesting work around this area of mixing and spraying, as are other groups as well. So please do look them up. So in terms of the device that we made, it was really home built. So you can see, it was made from wood from a cupboard. Actually, we got these syringe drivers, a lot of stuff that we can beg and borrow from the lab. We had these blotting pads up here, and we had a very rudimentary spray nozzle down here that would spray on a fast moving grid. When you turned it around, you could see, how Heath Robinson this was. We had a lunchbox to stop us being electrocuted, a pump from a fish tank, which went into these high pressure chambers to generate the humidity. It actually works really well to generate humidity very fast. But we had these sort of elaborate setups, but they weren't perfect because we were just trying to use what we could find sort of around in the lab and get these things together. But that we could do. This is the machine in action in real time. It's looping around. We wouldn't go up and down. That would be madness. It just goes one way. But it just gives you an idea of speed that the tweezers pass past the nozzle. And so bear that in mind, because in the next slide, we've slowed everything down. And this is one point where I really need to sort of emphasize why collaboration is so important, not just within fields, but between fields as well. So one of the key things that for us really moved this project on was going to Hamburg and speaking to the X-ray crystallography community. In particular, Martin Trebin, Dana Montero are in person, and many of you will know, they developed amazing XFEL, time-as-old crystallography expertise down at Hamburg. And so we went there for a few days just to chat to them, to sort of learn from them, learn what they were doing, learn some of the things that we need to be aware of. And it was incredibly informative, and it helped us out so much. We also managed to steal some of their technology as well. So what you can see down here is one of the microfluidic mixers. This was designed by Martin Trebin. It should spray crystals in a linear array, but we found that if you crank these up to 11, metaphorically, and really push them hard, you get a chaotic spray. And so what we can do is we can mix and spray. Our grid's shown here in number 1. The spray coming out is here. And then you can directly spray onto the grid. The droplets land on the grid. Then the grid goes in the liquid ethane. And then you can image this in the microscope. I won't bore you with the details. There's papers out there that show how fast the droplets go, how big the droplets go, but it allowed us to parameterize the setup. So we know that we can go from 5 milliseconds upwards using this approach. So from mixing to freezing, 5 milliseconds. And we've been trying to develop new machines. This is one of them. I'll come on to another one in a few slides' time, but we've had a number of people come to Leeds to try and sort of develop their own bits of kit. I think Hiroshi might be joining us from Japan, actually, who came last summer, spent 2 weeks in the lab. He's an amazing researcher, and he was able to take photos, use the kit, and then build a really cool system over in Japan. He's added his own adaptations, which have made it a really nice system over there. And so we are welcome to collaborations. People are welcome to come to Leeds, and we're more than happy to share the technology with you. But it is still can be quite challenging to put this together. We've now done everything via 4 labs where we can. So you have the breadboard, a camera now, so you can see the droplets coming out. We have a spray nozzle and the grid. The tweezers now go on an arch rather than plunging down, because this makes it a lot easier in terms of you don't need these large drops. It's a bit easier mechanically to control this. So we are now starting to make more modular, easy to use systems as well. Although, like I say, I'll come on to another system later on, which I think is probably where most of our effort is going at the moment. So we can use this to do a variety of different things. We've done small things like mycin, 100 kilodaltons. We've done viruses, ribosomes, apoferritin, because everyone's an apoferritin in EM. So we've done all these sorts of things. We can get down to 2.2 angstrom resolution, but you'll be mad to use this approach to generate high resolution EM grids. If you want really good quality grids, then you use the Vitrobot, you use the Vitrojet, you use the Chameleon, those sorts of machines that are designed to make good EM grids. This is really designed to do fast mixing, time resolved experiments. It's not about high resolution, but it does still get to modest resolution sort of EM structures. And so I'll just give you one quick example of some of the things we've been able to do. And this first one is myosin. So these are one of these videos you can find on YouTube that are sort of bedded in reality, but there's a few sort of artistic licenses given. But fundamentally what we have is a motor protein that can run along an actin track and deliver cargo. And this is a protein that's been incredibly well studied by a number of groups, not least the groups over in Leeds as well. So what you can see running here is a movie of myosin. This was done in negative stain, and this was done probably about 20 years ago now by Stan Burgess, who's an amazing microscopist, along with Matt Walker, John Trinick, and Peter Knight. And what they were able to do was to take their 2D classes of myosin and actin and put them together in what they thought would be the most logical way, and then make these apparent movies of myosin running across the filament. And it gives a really good insight into the apparent sort of mobility of this system and the way that you have these apparent legs walking across the actin track. And since then, a number of groups have gone on to solve structures of all the different sort of states of the foot domain as it interacts with the actin via X-ray crystallography and electron microscopy. And so what happens is that you have the foot region on the actin track. ATP releases this. The ATP is hydrolyzed to ADP phosphate, and then you go into what's called a pre-power stroke, where the energy is stored in the motor domain. It then lands on the actin, the energy is released, and the arm region swings over. You then get this post-power stroke state, this tightly bound ADP phosphate state, and then the ATP comes and releases it again. And as I say, all of these states have been solved except one, which was this one here, which is the pre-power stroke. And this in many ways was the most important state, because at this state you've hydrolyzed the ATP, so you've got the energy in the system, but it won't release the energy. So it's a bit like pulling the elastic back on a catapult. You've got everything primed to go, but you've not quite released that energy until you hit the actin track. And we wanted to know what the structure looked like when it hit the actin track to release the energy in the system. And so the experiment was relatively straightforward, but why hadn't anyone been able to do this before? Well, that's because when you add the ADP phosphate form of myosin to actin, it translates to the pre to the post power stroke in about 100 milliseconds. So it's really hard to capture this via standard approaches. So a simple plan was developed. I say simple, the idea was relatively simple, but it actually started out in the late 1980s when Howard White and John Trinick first sort of came about doing this. But it's taken that long for the EM technology and the time resolve technology really to catch up to be able to do these sorts of experiments that they were dreaming up over 20 years ago. We also used a mutant form that slowed everything down, which made life a little bit simpler. So we have our ATP, we mix it, we get the ADP form, then we add it to the actin, and then hopefully we can capture the state. So using the setup that we had at the time, we added the ATP to the myosin, and we left it for 2 seconds in the line. We then added the actin. We then sprayed this directly onto the grids in 10 milliseconds and 120 milliseconds. And the way that we changed the time was relatively crude but effective, which was that we simply just moved the distance from the nozzle to the ethane. So this is 10 milliseconds here, where you have a shorter distance to travel. By increasing the distance, takes you to 122 milliseconds. So to put huge amounts of work into one slide, we collected numerous data sets. We actually did triplicates of these experiments. And what was really nice was that when you look at the ratio of the pre to the post power stroke, the two states that we were looking for, you can see that this ratio goes down over time, as you would expect based on the kinetics. The pre power stroke state is going down, but also we got really good correlation between the ratio of the pre to the post power stroke in our data sets as well. So the mixing is actually pretty consistent between these different grids. So I don't really have time to go through what this means in terms of the biochemistry too much. Suffice to say, we were so happy to have finally, captured this prime state that, has been elusive for so many years, and something that Howard had been working on for, like I say, well over 20, 30 years to capture. So we were finally able to do this. And obviously, when you do these things and you get these different states, then the first thing you do is you make some macromolecular movies to sort of overinterpret the data to some extent. But that's being able to do this is really nice, because what we could finally see is what the structural rearrangement was that allowed the triggering and the release from the pre to the post power stroke. And a lot of this was about moving back this subunit, which you can see in pink, which is the upper 50. This then releases the arm to swing across, and you can then produce productive power stroke. But I think it's really important at this point, though, to mention that obviously the structures were really important in understanding what was going on in this system and how the force was transmitted, etc. But what was also important was just the wealth of data that was out there by other groups as well. So the biochemical data, the mutational data, the other biophysics data. And it was actually quite nice to go through all this other data, and a bit like a big logic puzzle, really, pulling it all together and then explaining data from, 5-10 years ago through these models, and using that data to build up this much bigger picture of what's going on. So the structure alone was informative, but actually it was once we started bringing together all this extra information, then we could really start to understand what was happening within this system, and this multi-technique approach really to understand these questions. Away from mycin, we've done a few other things as well. Some I can talk about, some I can't. We did actually start to look at virus decapsidation. This is sort of being written up as we speak. Hopefully we'll see the light of day one day. Intriguingly, we thought this would be milliseconds. It turns out to be nearer seconds. But what it allowed us to do was to start to capture the virus in different stages of capsid openings. We could start to understand the mechanism that this capsid undergoes in order to release its internal cargo. It's not just about squishy proteins either. We've been working with material science as well. We've been looking quite a lot at sort of nucleation and crystallization as well. Again, looking at how certain sort of structures form in the crystallization process. We've also looked a lot actually at membranes and how membranes get shredded up with different polymers such as SMALPs and amphipols and things like that as well. So some of it's protein based, some of it is more materials based. And so we have a system that works. One thing that you always get asked is, can you go faster? And that's one thing we were looking into. So we were limited by 5 milliseconds, which, would take, it's still pretty fast, but as soon as you're close to microsecond, you feel like you should go to microseconds. So we were looking at ways in which we might be able to do this. And the answer came from a trip to our collaborators in engineering. So what they're able to do is to write through liquids. So in the top left-hand corner, what you can see is a nozzle here, which is actually delivering a high-powered backflow of gas. This gas then dispenses the liquid and actually right through the cavity that the gas forms onto the surface of the dish here. And so we used this approach. We thought, well, what if we put our grid just under the liquid ethane? And then this is the microfluidic nozzle that you saw on the spray machine. What if we mix in the nozzle, and then as we pass over here, because we have a backflow of gas, it will displace the liquid ethane. That then allows the droplets to land and spread out and then freeze again, because you're forcing them onto a cold grid. And that's basically what happens. So this is actually on the loop. We don't just spin around 360 degrees. But what we do is we very quickly pass the ethane. You can probably just about see the ethane ripple here. So we mix in the end of the mixing chip, and then we spray directly on the grid. And the distance is just a few millimeters, which allows us to mix and freeze in the microsecond time scale. And so we wanted to test this out biochemically, and we did this with the actomycin complex. So actomycin plus ATP dissociates, and it dissociates incredibly quickly. So what we can see is after 3 milliseconds, we will get full dissociation at the ATP concentration we were using. And so what we did is we mixed ATP and apiferritin, just so we could tell that ATP was there. So we needed a fiducial marker effectively. And when we mix these things together, we see near full decoration of these filaments still. And so when we did the computational work, we found it's about 80% decorated. So we were at about 400 microseconds mix and freeze time, which correlates quite well with what we calculated based on distances, spray speeds, etc. So we can get down sub-millisecond. There is a bit of a health warning with this in that, it still work in progress, and there's still a lot of development to be done and optimization of these approaches before we can really start to push this forwards. We have still some other structures using this approach, but the ribosome and apoferritin are relatively straightforward to doing EM, and we're still working on some more challenging systems. But I thought at this point it's worth noting a few other people's work, just because I think there's some amazing stuff going on at the moment, and I think it's only fair to sort of point out some of the other sort of really cool time-resolved electron microscopy work that's going on. So one of these is from Chris Russo's group, and this was published just a few months ago in PNAS. Some of you may have seen this, and this is similar to what I've shown you previously, where rather than spraying directly on a grid which is just under the ethane, here what they do is they place their grid in the ethane. They then lift it out of the ethane, so you still get liquid ethane remaining on the grid. You then spray your droplets on the grid. The grid goes up, the spray turns off, and then you plunge your droplets back into the liquid ethane. And so the droplets themselves are freezing as soon as the grid passes the spray nozzle up here, because you've got the liquid ethane that resides on the grid. So you're getting freezing here. So you can imagine if you're mixing at the end of here to here, you have a very short distance in which droplets have to travel before they're frozen. So this could be a really neat way of, again, doing very high speed time resolved EM. So I do urge you to read that paper. Another one is the Ulrich Lorenz group, and they're doing some amazing work. And this is just one of the experiments that they're doing that I thought I would draw your attention to. And what they're doing here is taking advantage of the fact that your protein is frozen in ice. And so here what you can do is mix your protein with a cage compound in the ice. You can then use UV or whatever you want to decage your cage ligand. Well, of course, because it's frozen in ice, you can decage it, but it doesn't diffuse anywhere because it's stuck in the ice layer. But then what you can do in the microscope or in a separate stage is to melt the ice very, very quickly on the EM grid. This then allows diffusion to happen and mixing to happen, and then you can refreeze the ice very quickly. And this allows you to get to really low microsecond time scale mixing and freezing. It's beautiful work. So again, if you're interested in these sorts of things, I do urge you to look at Ulrich's work as well. I think they're both really interesting approaches to this, and it's all complementary rather competitive in my view. So that's going fast. One of the real challenges we have, bizarrely, is going slow. So there's two ways we can do this. So let's say we wanted to go at 500 milliseconds rather than 5 milliseconds. We could, for example, mix our ATP and protein, whatever we want to mix, spray it on the grid, and then just leave it on the grid for 450 milliseconds or whatever, and then plunge it. Or we could mix and then pass the solution through some tubing so it doesn't interact with the grid until the last seconds, well, the last few milliseconds when it hits the grid, and then gets plunged in liquid ethane. So one's mixing on the grid, one's mixing in flow. And you'd have thought that they would give you similar results, but they don't. So again, we're looking at the actin-myosin complex. So if you do this experiment with limiting amount of ATP, what happens is you add ATP, you get dissociation. But actually, you then get reassociation after around 500 milliseconds. But when you do these experiments on the grid, you don't see reassociation of the myosin. The myosin falls off, never comes back. If you do it in flow, then what you'll see is that the myosin falls off the actin when you add the ATP, but then it reassociates again. So you get reassociation in flow, but you don't on grid. And we think that's because what happens is very much the bogeyman of EM, which is called the air-water interface. If you imagine you have a very thin film on the grid, what happens is your protein dissociates, and rather than staying in the bulk liquid, it diffuses to the air-water interface and sticks there and gets sequestered away. So it can't come back to reassociate. Whereas if you do it in flow, there's no air-water interface, so it can reassociate again, so it's not stuck at the air-water interface. And these are things we really need to think about for time-resolved experiments, because what's happening in bulk is not happening on an EM grid. And there's more things that we need to worry about in terms of that sort of chemistry. So this is something that we did several years ago now. And this was where we were looking at protein degradation over time on an EM grid. So here, for example, you look at the ribosome, the full subunit or the large subunit. And for the L31 subunit, it's associated at 30 milliseconds and 54 milliseconds. But if you make the grids beyond 200 milliseconds, this subunit falls off. The S2 subunit has a stronger interaction, but still falls off somewhere between 200 milliseconds and 6 seconds. So something's going on the grid, which means that these protein complexes are starting to fall apart. And we think it's something to do with interactions with the air-water interface. Again, it's chemistry we have to think about in terms of time-resolved experiments and something that we're competing with. Something else that we found, and this is coincidentally, we're working on a protein called HSPD1 as a drug discovery target. And what we found was that when we made the grids on the Vitrobot, we had a disordered loop, which we didn't really think much about until we compared it to the bacterial homologue, where the loop became ordered. And then when we looked at the cysteine residue close to this loop, we could see it was modified. And then strangely, we went back to the grids that we made on the sprayer for a different experiment, and we could see that this loop region was actually fully ordered. So if you made the grids fast, you got an ordered loop. If you made them slowly, you got a disordered loop. And if you added DTT in, then you got an ordered loop. So it looked like there was some sort of modification of the cysteine residue in a time-dependent manner. And we've really gone down the rabbit hole with this. And this is just another example. If you go into EMDB, there's lots of examples. And this is beta-galactosidase. So here you have a cysteine residue. If you do these experiments on the copper grid, what you see is modification of the cysteine residue. On the gold grid, no modification. On the gold grid where you add copper back, you see a modification. And on a copper grid of catalase, you don't see a modification. So what's going on? Well, we think that the copper is leaching off the grid. And we've seen that coincidentally in a totally different experiment we were doing, where we made a protein on a copper grid and a gold grid just to see which would be the best. Both got to similar resolutions, but what we saw was that when we made the protein on the copper grid, we could see a metal ion in a binding site, which we assumed to be copper, which is leached off the grids. So we've got copper in the solution. There's also quite a lot of work out there from different fields that shows that in micro environments, thin films, you can get a lot of chemistry going on. In particular, you can get peroxide formed. And so peroxide plus copper equals a Fenton reaction, which means that then some of these cysteines are getting modified. There's a lot more work to be done, and I'm not a chemist, but there's certainly chemistry going on that we don't understand on these grid surfaces that we need to get to the bottom of. And there's 14 cysteines in beta-galactosidase. Only one of them shows severe modification. So it's not just all residues, it's those that are in a certain environment that allows them to be modified as well. But there's certainly chemistry that we need to be aware of when we're doing our time-resolved experiments. So we have a thin film environment on the grid. We can get preferred orientation, which I won't discuss today. You can get chemistry, which we don't fully understand, and you can get proteins falling apart. So we just need to be mindful of that when we're doing our experiments. And so this leads us to sort of one of the new machines that we've developed. And this was with two things in mind. The first was, well, if the time that the protein's in the thin film is important, then why don't we minimize this time as much as possible? And the other one was, how can we make time-resolved methodologies a bit easier to access to the wider community rather than having to, go on eBay and build these big machines? And so we developed a new machine over in Leeds, which we've called the Speed Blot, which you can see on the right-hand side. And this is effectively a blotting machine. And you can see here in the video, which is going on in a loop, that we effectively have these little blotting arms. They blot the grid and then it plunges in the liquid ethane. We can actually blot in as little as 20 milliseconds and still get structures. So we got about 2.6 angstrom structure from a 20 millisecond blot time. So we can really minimize the time that the protein is in the thin film environment for. But actually, the thing that I want to talk to you about today was actually the way in which we can use this to do time-resolved experiments. So hopefully you can see the video that's playing on the side. But if this isn't quite so clear, I've also got some stills from a different experiment that we did to sort of explain how we're doing these experiments. So what we have are two impermeable silica pads on the top here. And we have two droplets, one on each pad. These pads come together and they actually vibrate against each other to mix the solution. The pads then pull apart and then deliver the droplet onto the grid. The grid's then blotted and then plunged into liquid ethane. And you can see the blue mark on the pad here, which shows that this has been moved onto the grid. You don't get full transfer, but you get enough to make the grid. And so you can see the experiment here where we've done slightly more complicated because we've actually added something else to the grid, mixed two things here, added them to the grid, then blotted and then froze. And we can do all this in a few sort of 100 milliseconds or so. And so it's a really straightforward way of mixing and freezing. So back to our favorite system, actomycin. Here you have the actomycin complex. We add ATP, and you should see dissociation, and you do see full dissociation, even at 50 millisecond mix times on the mixing arms. Coincidentally, you also see betamycin in the background, which might be due to the fact that the protein hasn't degraded in the thin film environment, to a total time of 350 when you take into account block time, etc. We can actually go faster than that now, and this was very much low-hanging fruits. It's a very fast reaction, but we've done other experiments now to show that the mixing rate is pretty good, actually, about 80% mixing. Not as good as microfluidics, but much easier to do. Of course, one problem we've got is one that I showed you earlier on, which is that proteins get sequestered away to the air-water interface, and that's going to create issues when you're doing these sorts of experiments. So to highlight that, we actually tried to do the myosin and the actin mixing experiment, and what you see is that they don't mix. However, we can steal some ideas from the EM community, and one of those is the use of detergents. So people use detergents to cover the air-water interface to stop interaction with the air-water interface. So if you have DM, then what you find is you put DM in the mix, you then get mixing the myosin and the actin. So the myosin isn't being sequestered away at the air-water interface to the same level now. Therefore, we see these mixing experiments on the grid. We have done it as well to avoid aggregation. So here we've had an aphamer. When we add it to a protein, you see horrendous aggregation. But if you add it and then make the grid within a few hundred milliseconds, you can capture the binding event of the aphamer before you get to the aggregation event, the full complex. And another thing we've been in a lot of the mixing for is actually to change the pH, the buffer composition as well. So here we had a protein in really high PBS buffer, so the contrast on the grid was dreadful. But actually, if you then very quickly mix to replace the buffer with something that's a bit more amenable to EM, you can start seeing much better contrast on the micrographs. I mean, it showed us that the protein was too concentrated, so we have to go back, but at least we can see the protein now to optimize the experiments. So one thing, just to sort of circle back a little bit in terms of the cysteine oxidation story. So we were thinking that a lot of this is due to the protein being in the thin film environment on the grid. So we did an experiment where we blotted the protein for 100 milliseconds. We applied it to the grid, blotted it, froze it, and we still saw a modification. So we were a bit disappointed by this. But then we thought, what if it's residence time on the grid, not just time in the thin film environment? And so here what we did is we have a grid. The protein is applied to the grid using the top arms of the speed blot, and we have a 20 millisecond touch time. We then blot for 100 milliseconds, and when we do that, we don't see the modification of the cysteine residue. So it's not just about the thin film, which is the problem. I think residence time on the grid might also be a problem. So again, just something to think about when you're doing your EM experiments. So a really nice thing that we did with Kyprianos and Pejun down in sort of Oxford and EBIC was to do some tomography style experiments. So here what we did was we were mixing cells with a substrate. The substrate we added gold fiducial so we could see that they mixed, and then we're able to do single-sided blotting of the grid to be able to generate these really nice sort of micrographs, which then can go down the tomography line. I'm not involved with the biology, so hopefully that's underway at the moment, but hopefully they'll be able to see something really nice now that they've been able to do these mixing experiments in a really short time scale. Something else that we did down with EBIC as well, and this was just published a couple of weeks ago in IUCRJ, so do check it out if you're interested. And we loaned them a unit, and they, with our help, took to it. They made their own sort of 3D printed arms, which are slightly extended. And the reason for doing that was that here you have a humidity because the droplets in the air for a bit longer than normal. They're triggering reaction with blue light. They're using spectroscopy to monitor what's going on the grid. And then when they're ready, they're blotting and they're freezing. And so it allows them to do inline spectroscopy of the protein on the grid and then to freeze at the appropriate time based on that. So it's a really neat sort of development of what we've already done with the speed blot. And so what I really wanted to do is to put a bit of a call out as well. If anyone's interested in developing new modalities, do please get in touch. We're looking at sprayers for this, automatic deposition, faster motors. We're looking at laser traps as well and using light triggered reactions, which we're going to be doing over the summer. We've got a group joining us next week that are coming down that have got some really interesting time resolved experiments that they want to do. And we're developing bespoke ways in which they can do that. And I'm really sort of excited for that. It's going to be a really good few days where we're just going to build some additional functionality, change the programming a little bit for them, and then hopefully be able to do these time resolved experiments. So we really want to try and develop this as much as we can as like a community effort. Everyone has slightly different experiments that they want to do, and everyone has experiments that we've not thought about. So if there's something that you think we might be able to help you with, then please do reach out. We're really keen to collaborate with people and find new ways of, now we've got this base unit for sort of manipulating grids, to use it in a way that we can do more time resolved sort of experiments. And so with that, I'm conscious my time's nearly up as well. Hopefully I've shown you, time resolved EM is really important. It can allow us to explore a variety of different timescales, from microseconds to seconds, and length scales as well, typically from 50 kilodaltons upwards. But of course, what that does is leave a massive hole of things that are underexplored. So things smaller than 50 kilodaltons, that's where we really need lots of other additional technologies, which is where crystallography, HDX mass spec, for example, really comes into play as well. And of course, the faster timescales I think are going to be really hard by EM. For those longer timescales where maybe crystals start to crack, etc., we can start to take over from there. So I think there's so much collaboration that we can do. And I think it's just an excellent time to be in sort of the time resolved field at the moment. There's so many complementary techniques out there that we can use to sort of answer these questions. And the questions that we're trying to answer now are getting more and more complicated. And I think it's so hard to do it with one technique alone. So the ability to integrate this to other techniques, we're really keen on. So again, I'm really happy to hear people that want to try to integrate this. How do we integrate the data that we get from the XFEL beamline into our EM experiments, into what we're getting from our HDX mass spec experiments? AFM, for example, high-speed AFM gets ridiculously nice sort of data back, looking at proteins non-frozen in native states in membranes. How can we integrate the high-speed AFM data with what we see in the EM or the NMR, etc.? So I think there's a lot to be done as a community, and particularly between the CCP programs as well, of how we can better integrate the data that we're getting back from these different sort of experiments. And so with that, just a few thank you. So massive thank you to Howard White, who I first met as a postdoc, and has been a mentor and above anything, a friend really, for so many years, developing these time resolved applications. And none of this would have been possible without his sort of enthusiasm and sort of help over the years. The EM facility are amazing. Becky for the early work on the sample preparation stuff. Louis and Charlotte have helped a huge amount with the speed blot as well, as has Nick, who's been absolutely instrumental as the engineering chief who's been developing the speed blot with me, and Nick, who's been involved with the microsecond spraying. Charlie, who's just a brilliant researcher, and Sean as well, of all the stuff they've done around the mycin, which has been amazing what they've been able to pull out from the data. David Clable, who is just a ridiculously good PhD student who did all of the mycin, or pretty much all the mycin data collection stuff, and so much of the early time resolved work with me, and Izzo as well, brilliant PhD student looking at the protein modification, and everyone for funding. And thank you for listening.
Okay, thanks very much. That was great. Got a few nice clapping hands floating up.
Okay.
Great. So we have a couple of questions, if you're ready. Alexandra Svejin says, actually, I don't know if we can unmute and ask the question yourself. I'm not sure who has permissions. Otherwise, I will read it out for you.
Yeah, if people want to unmute, they should raise their hands and then we can make that possible.
Okay.
Or you can use the chat.
Okay, well, I'll just read it out for now to stop. So, great talk. I'm interested in the cyst modification that you were investigating. Is there a publication or available structures showing these modifications in more detail?
There is. There's one publication, which we managed to get away with the title, which started with a CIS, which was in PLOS 1 a couple of years ago. So we did that as a special issue on EM. So if it was with myself and Yiheng Wang, David Clable, and Becky Thompson. We've not yet published all of the data. We keep going down rabbit holes of new experiments to do, but I think we're nearly there now with the next set of experiments. In particular, now we've got the speed block being able to show whether it's residence time on the grid or time in the thin film. What has been really interesting is that I mentioned this at CCPM when I gave a talk a few years ago, and I now have an email inbox filled with problem cysteines from various different proteins that people sent me. So it does look like it is quite a thing in the field. And it's that usual thing, you notice it in your map. You don't really want to mention it in your manuscript because then the reviewers are going to ask you to spend 10 years trying to explain what you're seeing. And so I think these things have gone a little bit under the carpet to some extent, but there does seem to be quite a bit of evidence when you go for the EMDB for these sorts of modifications as well. I think coincidentally as well, they might be quite interesting because for HSBD1, I think that cysteine senses redox, and not all cysteines are modified. And I think it might be those cysteines that are particularly responsive to things like redox that have been modified. So it might be giving you some information around that as well.
Okay, thank you. So, where's the name gone? There we go. Herang Huang says, thank you for the great talk. Do you think intermediates identified by time-resolved EM also need biochemical validation, or can the structures themselves be sufficient to explain the mechanism? Also, I would love to have a speed blot in our lab. Roughly how much does it cost compared to a VitraBot?
It costs less, I can tell you that. That's the good news. So send me a separate email. I don't want this to be a sales pitch. If anyone wants to know about that, I'm happy to send you quotes and other information about that. In terms of intermediates, I think the more information you can have, the better. I think for any sort of experiment like this, you've always got to be careful with what you're seeing in EM and how that then relates. We always make sure we go all the way to completion, so we're not going off pathway. But where you can, I think additional biochemical data always helps. The EM will get you so far, and it can get you quite far. But from our experience, it's always nice to have additional data where you can, try and lock different states, or, have a HDX or something like that. But again, it depends what you have access to as well.
Thank you. So from Sophia Diletta, sorry, it's truncating the names for me. I can't get the from Sophia. Thank you for the interesting presentation. Regarding data analysis, which are the biggest computational challenges when trying to sort through the continuous heterogeneity of a time-resolved data set? And do you need to collect significantly more data than under standard conditions?
Yes and no. I mean, it's always a bit protein dependent. I think what I would always recommend, and this is why I tried to mention during the 2D, sometimes it's about doing the histograms. And where you get a really nice experiment is where you see that at start point A, you see one state dominate. You don't necessarily need millions of particles. You just need to show that 95% of the particles go to a certain state. But then over time, you can see that state isn't in your data set anymore when you've made a grid at, say, 50 milliseconds and 500 milliseconds and 1 second, whatever the time points are. But you can see other states start to appear. And then that allows you to have some confidence that what you're seeing is sort of for that particular time point. I think one thing it'd be challenging to use time resolved EM to do is to say, well, that state must be at 53 milliseconds. Yeah, I don't think you'd want to put time on it, but I think what you can show is trends of states appearing and disappearing over time. And I think in terms of data processing, some systems are really amenable to this. So one thing we were looking at was a doddle, because it was two things coming together and then a large rearrangement. And that was really clear in the 2D classifications. If you're looking at really subtle rearrangements, that might start to get really challenging of how you start to pull those apart. So you don't always know what it's going to be as you're going across as well. So for the myosin one, it was quite easy because we had a lever arm that went from the pre-power stroke to the post-power stroke. So there's a very clear conformational difference between the two that we could get at via the 3D processing. Although we did spend a huge amount of time trying to find other states, and we never did. And that's probably just because they're low population in terms of numbers, or just the transition so fast we couldn't capture them. But yeah, it's a tricky one because it's quite sample dependent.
Yeah, I was was going to comment on the for the myosin pre-power stroke example. You had 3 grids, like 3 replicates, which were amazingly close together in terms of confirmation populations. It seemed, yeah, I mean, it seemed almost too good to be true.
It did.
That
No, we
level
were
of
really
agreement.
surprised. We were a bit worried by that. It was almost too good. You know, it'd be nice if it's a bit more variable. But yeah, I mean, just for this, I suspect it's probably because we go, it's quite integer, one to the other. And so I think that probably helped. If we had 3 or 4 different states, I suspect then you might have seen more variation. But I think because of that, it was a bit easier for us to characterize it and capture it as well. But in terms of the mixing and the speeds, then we actually have those quite well down in terms of reproducibility with the mechanism that we have.
Okay, right, we've got a couple of hands up. And I can't see it. I can whoever's number 1 hand. Sorry, I can't see your name. No, okay,
Gary.
it's got this is the So yeah, please, if you unmute, I think you can talk.
Hi Steve, how you doing?
Hi Gary.
Nice
not about
talk.
NMR, is it?
No, I promise you it's not about NMR.
That'd be a nightmare.
I got the indication that you didn't blot on all of these ones. Do they all get blotted, or do some of them get blotted? And the follow-up to that was, if they don't get blotted, could you sort of have it so that it's pre-frozen in ethane, pop it up, spray it, and pop it back down again to get something really fast?
Yeah, no, there's some really good points there. So in terms of the way we spray, we don't blot, we just spray and plunge.
Yeah.
For the speed blot, we always blot and go in, although we're looking at spraying and blotting. But you're right, and it's a really neat trick that I've seen others do, where you could freeze your grid and then pull it back out again, and then spray a reactant on the ice, plunge it. You go as many layers as you want to make a little sandwich of reactants. And then when you devitrify the ice, you then get the diffusion through. And I know other groups looking at this, because I think it's a really neat way of maybe doing these experiments. The challenge is going to be getting that layer which is quite universal across the grid, but I suspect that's a problem that can be solved.
Yeah, very cool though.
Thanks.
I hope you hear me over all the background noise. I'm in a mass
spec
spec lab at the moment.
So I can see Sarah's hand up, but if I could just go to another question from the Q&A, which seems to follow on very nicely. So from Leo Sazonov: great talk. Droplets from spray can form quite thick drops of ice on the grid. Did you find the ways to improve spreading of drops? So I think that's just what you were.
I think the fact that we're blotting tells you some of the answers you need to know with that one. We managed to get it working a bit better, but we never managed to crack it. I know other people have. So, the NCHEV group at the CRIC, I think they get much better droplet spreading on their grids, and I think others have as well. We've kind of moved away from that a little bit, because we were doing things like the hairy grids which you get on the chameleon. We were using those, lots of glow discharge, different buffers to try and look at the spreading. So it's okay, but it's not perfect, and I think there's a lot of parameters you need to get right for that. And so that's one reason why we're moving a little bit more towards the sort of blotting approach now at the moment. But I think it is achievable. One thing which I didn't go into, which is a conundrum I think the field needs to tackle, is that when you spray, the faster you go, the lower the concentration of protein you see on the grid is. And so if you look at Chris Russo's paper in PNAS, for example, I think he was using something like 40 mg per mil apoferritin, so that 40 times the amount of protein he would have done had he used a Vitribot. And we saw the same thing for the sprayer, where once you start to get to 5 milliseconds and 10 milliseconds, you start to need an order of magnitude more protein. And for certainly people coming with membrane proteins, they were in tears when you're like, well, actually, if you could just make, 10 times as much of your really delicate GPCR, please, that'd be much appreciated. It's not practical. And that's another reason why we've moved a little bit away from the spray approach. Again, I think there's ways around it, but at the moment, that's another hurdle that we came across with the spray approach.
Thank you. Sarah, would you want to say hello?
Hey.
Hi, Steve. Lovely to see you. Thanks for an amazing talk. So you showed the lovely animation of myosin walking along the track, and you also said about other tools that might be integrated with the time resolved EM, but you didn't mention simulation. Do you think there's any room for simulation to make a contribution?
Of course there is, Sarah. And I think it's going to be so important to bridge those gaps as well. You know, EM's always going to be able to, it'll salami slice to a point, but there's always going to be gaps in between those states that we get. We're never going to be pulling out all those different and that's where I think, because the problem with the MD sometimes is trying to go from here to here is just too big a gap to bridge. But if we can get 2 or 3 intermediate states in between there, then the MD will beautifully bridge between those gaps. And I think that'd be, yeah, I definitely, apologies for not mentioning it more, but absolutely there's a role for MD to sort of play in bridging those gaps between it. And I think this is where HDX Mass Spec and others come in as well, because they can give that really much finer slicing, which I think could be really nice for sort of bringing in those bits of information that we can then use. And whether there is, I mean, the idea that we have this computation actually at the heart of it. And that might be a way that we can integrate all of these different technologies as well, is if actually the computational model is in the heart, and then you put the EM and the mass spec and the NMR and the X-ray data into that, and that's then how you build up on it. I mean.
Right, I think we've got one more question in
Thanks,
Q&A, and then I'll come to John. So Evandro, congratulations on your amazing talk on time resolved EM. Really enjoyed it. I was wondering if you have any plans to use this system for cryo-trapping experiments in macromolecular crystallography. It seems like it could have some interesting applications too.
Yeah, absolutely. And you're right. I mean, if you're interested, do reach out. But actually, we do have collaborated with a few groups at the moment where, you know, for exactly that reason, because we can put, well, for micro ED, one thing where we have the microcrystals on the grid, but also potentially for just loading the crystals in the loop onto our machine as well, and then integrating some of the technologies we have across the two. So the X-ray crystallographer, you know, that I was trained as is still there. And so I am still sort of looking at ways in which we can bridge both of those, because again, it'll be quite nice if you could have a piece of kit that could cross between those two disciplines. So I think there are ways we can do it, but if you have something in mind, do reach out, get in touch, because I'd be happy to chat. You're welcome to come to Leeds and we could see how we could integrate it, because there's loads of different experiments that people want to do. So I'd be happy to talk to you about that. Yeah.
Okay, John?
Thank you very much, Stephen. I thought that was really brilliant.
Oh, yeah,
and I learned a great deal. Just 2 points on a compare and contrast with the crystallography, and you made points about that. One big advantage, it seems to me, that the single particle has with the laser flash is you don't need the laser powers that the crystallography needs. I mean, a big change between the synchrotron and the XFEL was the ability to work with a smaller crystal volume, and that reduced the power need as well. But you, seems to me, you're totally free of anxieties and need to do a power titration, which is needed in the crystallography. And Ilma Schlichting has done a good deal of work on this and really sort of put us into the reality check, if I may put it that way. But then the second thing, perhaps where the crystallography has the advantage, which you didn't mention, is estimating coordinate error estimates.
Yeah, absolutely.
And so I found this approach in the CryoEM where you split the population into 2 halves, and then you do a full analysis on each half, and you get a coordinate precision that way. And of course, the disadvantage is you're halving the population size for each analysis. And then at the end, of course, you can recombine those to get a coordinate precision. But it's not been, as far as I can tell, widely adopted. Anyway, I just thought those 2 points on the compare and contrast were rather interesting.
No, I think they're important, and validation is going to be so important, because the last thing we want is things going wrong. I suspect with EM, it's going to be looking at larger changes as well. I think where crystallography is going to come in is looking at those fine differences that you see at those high speeds. I don't think EM is necessarily going to be looking at those. I think we're more for the large domain motions, watching machines ratchet across, where the precision maybe it's a different sort of ball game in terms of what we're after, I guess. But I think that might be something that will come in. But you're right, validation in EM at the moment is an issue, and I think it's something that we still need to get on top of.
Yeah, and I think just very briefly, the seeing change and believing it is two different things, and we have to get a proper handle on these error estimates. In both approaches, in the crystallography and in the CryoEM, because I'm sure you'll come to the point where you think you might see something subtle rather than large movement, and you're excited about it, and then it's going to be all about estimating the coordinate errors.
I mean, it comes back to that earlier question, doesn't it, which is a very good one about the biochemical data to back it up. And I think that's the thing, isn't it? It's like, okay, well, now I think I know what's happening. How do I design the experiment to back up what I've seen? Is it fret pairs? Is it some sort of mutational analysis? What do I need to do to sort of prove what I've seen? And I think it's really important, because like you say, seeing isn't always believing, particularly in low signal to noise data.
Yeah. Anyway, thanks ever so much again. That was really great.
You're welcome.
Okay, great. I think if I haven't missed anyone, I think that was all of the questions. And yeah, I'm aware of the time, so I think we should probably wrap up there. A couple of meetings were put into the chat. If I can just flag them up again. DRIIMB has its own integrative structural biology conference in October in Leeds. Absolutely well worth going to. And there's a CSP Biosim training week at the end of September. There are URLs in the chat if you're quick.
Yes.
So I think
and the
it just
spectral
leaves me...
biology conference also has a workshop at the end of it as an optional thing. I'm not sure the registration page is quite caught up with that, but there will be a 2-day software carpentry workshop. So if you're doing computational work, you can make your software brilliant, as well as scientific.
Okay, thank you, Sarah. So if there's no other announcements, I think it just leaves me to thank Steve again for a fantastic talk, generated lots of interest. I think you're going to be getting lots of emails afterwards asking how much for this, how much for that. And if you're happy to loan your equipment out, which you seem to be, then that's
yeah,
great.
like I say, he's trying to get the science going, is it? That's what I'm really excited by, is the science. So just for anyone, if they're interested, just get in touch. And thank you for the invitation as well. Got to get back to my emails after this, which is not so exciting, but this has been great fun.
Okay, right. Yeah, okay, thank you for that. And we'll see you all next time. Bye.
Thanks. Bye.
Yeah.